Hi, so we're here at the 89th Annual Symposium at Cold Spring Harbor. My name is Melina Casadio. I'm an editor at Nature Cell Biology and Nature Structural and Molecular Biology, and I'm joined by Claude Desplan, who's a PI at NYU, and we're here at this beautiful Cold Spring Harbor location for the meeting. And so Claude, thank you so much for joining.
My pleasure.
So your lab has made fantastic contributions to our understanding of neurodevelopment and neuronal identity definition and optic nerve and optic lobe development, but you also study aging and rejuvenation. So what motivated this interest?
So actually, I've been working on flies for many, many years, but every time I work on flies, I try to figure out whether we can expand what we find to other model systems. So for example, when I was working on the early embryo on beacoid, we tried to find a species which could do the same thing as a fly embryo without beacoid. When we tried to work on color vision, we did find some nice thing on color vision in fruit flies, but flies are not very good at color vision. So we moved to butterflies to try to find something. And then at some point, I became interested in ants, mostly because I was contacted by my colleague Daniel Reinberg, who is a king of epigenetics. And at some point, it was like maybe 15 years ago, Harvard Hughes was trying to push his investigator to try to something completely new. And if you study epigenetics, ants are the perfect model because you can make some species up to 9 casts out of the same genome, and therefore he was basically trying to figure out what happened. But he didn't have much expertise, so he tried to build a consortium of people who could help him and us to basically develop the new system of ants. So again, there is him and there is Shirley Berger, who is also here, and a bunch of other people. And together we start putting together a model on an ant called Harpegnathos. And the reason we call this Harpegnathos, which is a large jumping ant from South India, is because they have a very specific feature that in general, in all kinds of bees or ants, when the queen dies, the colony will die because she's the only one that can reproduce. But in our species, when a queen dies, in fact, the young worker have ovaries, they have spermatica, and they can try to replace the queen. They only do so after a duel, a fighting, and after a while, 3 or 4 of the winner are going to become what we call gamergate, which are pseudo queen. They can completely replace the queen. Okay? And as a consequence, in fact, all our colonies are run by gamergates, by those former workers. They were born in the skin of a worker, and somehow, because they won the duel, they got promoted to becoming reproducers. They can produce eggs, and they can basically behave as queen. They stay with the nose up, and they basically don't do much of the work except laying eggs. Their brain shrinks actually also. And actually, the process is even reversible in the sense that if you bring a real queen into this colony, the gamergate is no longer able to maintain the colony. It's going to go back to work, and so this is basically an amazing phenomenon because for us who are interested in genetics, if you can make any organism becoming a reproducer, you can alleviate the problem of having a queen, that only one queen to make genetics, and you can basically make normal genetics. And actually in our system, we can make any kind of cross, not easy to make mutation, but you can make any kind of cross, and like in the fruit flies. But at the same time, we discovered something quite interesting, actually, that in fact, when a worker that was born to live for 7 months becomes a gamergate, is solo queens, her longevity becomes the same as that of a queen. And remember, a queen and a gamergate and a worker are exact same animals, except the queen during her development was fed some very special food. When she grew much larger, she had wings, and she also was able to have an ovary and lay eggs. The gamergate doesn't have wings, is much smaller, and it does have an ovary, which is a crest. And what's going to happen actually is that as the worker gets promoted to becoming a pseudo queen, she starts laying eggs, and at the same time, her longevity goes from 7 months to 4 years. Amazing. It's not 7% we heard like this morning. It's a huge extension of the lifespan. And once this gamergate is reverted to becoming a worker because somebody stronger came, longevity goes back to 7 months. Okay? So it's an amazing system to study longevity, and so we say there is no way we can let this go. We have to study the process. So again, this is quite amazing as a system, and really we could advance. So of course you have a limitation. You work on an animal which has very poor genetics, But nowadays with CRISPR, but mostly with genomics, you can study quite a lot of things. And also when you find something of interest in ants, you can always transfer it to fruit flies, for example, and study whether the process occurs also in fruit flies. So all the mechanistic aspect can be transferred into another system. So it's a place for discovering things that you cannot find in a normal system. So you use those non-model organisms to investigate something that is very unique to those, where this process can only be studied in this type of system. And ants, I think, is a perfect system for that. At the same time, when you investigate something, you discover a lot of very new things. And so if I have time, I can tell you more about, for example, how, indeed the fact that the dual gets promoted is basically when you lose the queen, it's because you lose the exposure over the worker to the queen pheromone. The queen pheromone repress all the workers, say, you get quiet, I'm the queen, don't move. When you remove the queen pheromone, and then this triggers the dueling. And therefore, we are very interested in figuring out how queen pheromone can be detected and how, when you have this dueling between two ants, they are going to go to basically try to show dominance by simply boosting up the level of queen pheromone and also lowering the level of sensitivity to the queen pheromone, and the opposite for the loser. And so, it's a lot of phenomena that you can basically find very interesting and understand how you can study the process. And again, it's not a model system, but there is a lot of things you can do nowadays on non-model system. And I think this is actually opening a lot of doors because, you couldn't even imagine how to do that before when, a non-model system was something you could not touch. So.
Right. Yeah, was the genome of that ant species sequenced when you started, or did you have to build a lot of tools?
Sequencing the genome is not an issue.
And you're able
10 years
to
ago,
manipulate it.
yeah. So, and genomics, it's very easy. We have a very good genome, we have very good transcriptome, and again, you can do, and all the technique, single cell sequencing is absolutely available there. You can do any kind of immunoprecipitation or ChIP-seq, or almost every technique you use in any model system, in cell line, you can study in those system as well. And actually, we take advantage of that very well. So maybe in my talk on Saturday, you will see that we can indeed take advantage of that. And again, it's the same as in fruit flies or in mouse. So,
right.
Yeah.
That's really exciting. And that reversible aging or the control of longevity phenotype that changes with the role of the ant is really fascinating. And have you found what analyzes
again, now we have a system where we have an animal that was born to live for 7 months. Yeah. And the animal remains the same. It remains the same skin because it cannot be moved. Genome. It just grows, same genome, but also the same shape. Yeah. It just grows a bit of abdomen to grow an ovaries, and they start laying eggs. And the other issue is actually normally, so this animal, as he wins the duel and start growing an ovary, is going to live for much longer time. But as you know, longevity is overall anti-correlated with reproduction. The more you reproduce, the younger you die. And here we have example of ants that live for 20 years and lay 1 million eggs. One ant lay 1 million eggs and she lives for 20 years. So again, all the concept, longevity and reproduction anti-correlated do not all that all. So there is something there. And actually we believe we discovered what's going on there, okay? So again, so okay.
So mechanistically, how do you think this works?
So again, you know that the reason why, very simplistically, it's believed that the more you reproduce, the younger you die, because the more you reproduce, the more you have to eat. Your insulin goes to the roof, and therefore you are going to fail to have the longevity, which is in part mediated by nuclear localization of the FOXO transcription factor, because the insulin pathway repress FOXO nuclear localization. Okay, but the point that the insulin pathway is actually a branch pathway. It acts through a receptor tyrosine kinase, through MAP kinase pathway, and it also acts through AKT and the FOXO repression. Okay. And so what we showed actually is that indeed the MAP kinase pathway is required for making eggs. If you don't have the MAP kinase pathway, you don't make eggs. And if you inject insulin, you do make eggs, but you can block it by blocking the MAP kinase pathway. But for some reason, in those ants which have very high level of insulin and a very high activity of the MAP kinase pathway, the AKT pathway is totally downregulated. That means FOXO is actually nuclear when it should be cytoplasmic because it should be repressed. And so we couldn't figure out what was happening there. And so we thought about it and we said, okay, what happened? When an ant wins the duel, it starts producing a lot of insulin, its ovary develops, So let's look at what happened in the ovary. And we discovered, in fact, that the ovary produce an anti-insulin called IMPL2. It's called IGF-BP7 in mouse. This anti-insulin does not block completely the insulin pathway. It only blocks the AKT branch of the insulin pathway, not the MAP kinase branch. And as a consequence of that, whenever insulin goes up, it produces insulin. Insulin leads to the production of the MPL2. MPL2 completely doesn't touch the MAP kinase pathway. You get the eggs, but it blocks the aging pathway AKT, and that's why FOXO is actually nuclear in this sense. So we believe maybe it's a very simplistic interpretation, but I think it's something we observe. So when we try to see whether we can transfer this into flies, and for example, overexpressing or removing IMPL2 in flies, expressing in the ovary what it should be there, and to see whether we can extend longevity. We're also making a mutation IMPL2 in ants to see whether you can prevent the longevity, extreme longevity. So again, this is a bit more difficult, but now it's becoming possible because all those technology is available now in them. Again, as long as the mutant is not early lethal, because we don't have conditional mutant. Yeah.
That's very exciting. And with these phenotypes that are so strong in ants and that lead you to new biology, what are you most excited to look at next?
Okay, so again, the mechanistic aspect, I know I'm not a biochemist. I was at some point, but I'm not a biochemist right now. My lab is not a biochemistry lab. And so we try to figure out, using, kind of genetic and stuff like that in mouse to see why you can block one aspect of the pathway, not the other. You can do, phosphoproteomics. So a lot of technology exists, again, in ants and in flies that you can use. And so try to understand the mechanism will be great. But at the same time, we also got very excited by the fact that, all of this event where the worker becomes a gamergate is triggered by the fact that she doesn't get exposed to queen pheromone. And ants are actually very, very olfactory animals. They rely a lot on olfaction because they spend a lot of time in the dark, and they have very tiny eyes, but they have very huge antenna, which they use actually to battle during the duel, but also the huge antenna which contain many, many types of olfactory receptors. They have a huge repertoire of olfactory receptor, more than we do as humans. Okay? And as a consequence, we have to figure out how you can actually figure out how basically ants can perceive those odors and discriminate with those odors, and how they can basically obey to the general rule of sensory reception, that for sensory reception you can only put one receptor gene per receptor neuron. So one olfactory neuron per receptor neuron. And for example, to put only one gene that can detect one pheromone into one specific. And if you have 400 genes like we do in fly, it's not an easy task. It's very, very difficult in ants to figure out how you can do that. But I think we solved it. So, and I will talk about it on Saturday. So, it's basically taking advantage of what happened in the fruit flies olfactory system, where you have a very deterministic pathway by which each olfactory receptor neuron knows exactly what it should do and which olfactory receptor it should express. Same thing happened in ants, but instead of expressing one olfactory receptor, it's expressed a whole array of olfactory receptor, 50 of them, okay? And then a second step, which is what we discussed, is able to choose which one of these 50 is going to choose in every receptor. So I think we solved the issue. And so it tells you, you can use those animals, and you just explore and you find amazing things that you could never find in other species, and you can explore them, and you have all the tools now to be able to do that. So it's a very exciting time for us. Because CRISPR is there, but it's not the most important. Really, genomics and how you can investigate in detail the mechanism is something that is really allowing us to progress at great speed. So
that's really exciting. Thank you for sharing, and I look forward to your talk now. I want to learn even more. This is really cool. Thank you so much for your time, and thank you for joining.
Okay.